The first step in translation is the process of initiation. What happens in initiation is that the mRNA binds to a small subunit of the ribosome and moves along it in a 5 to 3 direction until it reaches the start codon, which is the AUG codon. Then tRNA or transfer RNA gets attached. First, it attaches to the small subunit at the P site, and then it goes through mRNA until it lands on this AUG start codon. Then the large subunit will attach to the P site. The next step in translation is elongation. What happens in elongation is that the second transfer RNA will bind to the A site that is complementary to the second codon on mRNA. The amino acid carried by tRNA in the P site transfers to the amino acid in the A site because of a condensation reaction. The growing polypeptide then increases in length. Then ribosomes keep moving along the mRNA in a 5' to 3' direction, and the tRNA in the P site is then moved to the E site and then released. And the tRNA in the A site also moves to the P site. Also, one other step in elongation is that another tRNA binds complementary to the next codon on the mRNA and binds to the A site. Now, moving on to termination, which is the final process in translation. The termination occurs once the stop codon is reached, and once the stop codon is reached, or the termination point, termination sequence, then translation is stopped. These codons will recruit a release factor to attach to the A site, and then the polypeptide chain is released, and ribosome dissembles back to two subunits.